My name is Chad Hamilton, and I'm a practicing gynecologic oncologist and cancer researcher in the Washington, D.C. metropolitan area. I'm pleased to be here to provide this lecture and talk on the biology and genetics of uterine cancer on behalf of the Globathon, the Walk to End Women's Cancer. Endometrial cancer is the most common gynecologic malignancy in the United States and the fourth most common cancer in women. The American Cancer Society estimates annually about 47,000 new cases will be diagnosed, with an average lifetime probability of developing uterine cancer of about 1 in 38, or 2.6%. Just as cancer is not one disease but many diseases, uterine cancer is not one disease but many. We call them all uterine cancer because they share a fundamental feature, the abnormal growth of cells. Some of these cancers form in the muscular wall of the uterus. We call these sarcomas, but the majority, and most of what I'll be discussing relative to uterine cancer, develops in the lining of the uterus and is called endometrial cancer. To briefly review the clinical presentation and treatment, 90% of women present with abnormal bleeding in the context of endometrial cancer, and about 75% of these will present with early stage disease. We think that endometrial hyperplasia is the precursor for most endometrial cancers, and risk factors are usually associated with exogenous estrogen exposure. Surgery is typically recommended for treatment and planning, and additional treatments may be required for higher stages of disease. Before we get into a further discussion of uterine cancer and endometrial cancer, I think it's helpful to look back at some of the basics of cancer itself and of normal biology. Cancer, we now know, is a disease caused by the uncontrollable growth of a single cell. This growth is unleashed by mutations, that is, changes in the DNA that specifically affects genes that incite unlimited growth. In a normal cell, powerful genetic circuits regulate cell division and cell death. And in a cancer cell, these circuits have been broken, unleashing a cell that cannot stop growing. The secret, therefore, to battling cancer then is to find the means to prevent these mutations from occurring in susceptible cells, or to find means to eliminate the mutated cell without compromising normal growth. The challenge, though, is that malignant growth and normal growth are so genetically intertwined. The genes that disrupt control of normal cell division are not foreign to our bodies, but rather mutated, distorted versions of the very genes that perform vital cellular functions. To better understand the biology and genetics of uterine cancer, it may be helpful to review normal biology, then what goes wrong in cancer, and finally, what we know about what goes wrong in uterine cancer. First, for the normal. Through elaborate work done in the mid-1800s to mid-1900s, the molecular workings of normal biology were clarified, like placing pieces in a puzzle. The eggs of salamanders revealed that colored bodies, or chromosomes, duplicated when cells divided. And from sea urchins we learned that these chromosomes carried information vital for the proper growth and development of cells. Recall in 1860, the Austrian monk Gregor Mendel and his pea plants taught us that organisms transmit information from one cell to its progeny in packets of information that would later be termed genes. In similar studies of ancestry, fruit flies later revealed that genes were carried on chromosomes, and bacterial colonies later demonstrated that DNA was the chemical that carried these genes. So what does a gene do, and how does it do it? Well, we knew that at this point the genes carried information. From there we learned that genes carried instructions to build proteins, which are in fact the complex workhorses of the cell. Proteins carry out the bulk of cellular functions. They speed up reactions vital to the life of the cell, transmit signals from one cell to the next, form cellular scaffolding and structural elements, and they also, probably most importantly, regulate other proteins, creating complex circuits inside the cell responsible for coordinating the life cycle of the cell. A gene works by providing the blueprint to build a protein. A protein, in essence, is a gene realized, the machine built from the gene's instructions. But proteins are not created directly out of genes. An intermediate step is required, in which an RNA working copy of the genetic blueprint is created. It is through RNA that a gene's message is translated into a protein. So, genetic information is transmitted from a cell to its progeny through a series of discrete and coordinated steps. First, genes located in chromosomes are duplicated when a cell divides and are transmitted into progeny cells. Next, a gene in the form of DNA is converted into its RNA copy. And finally, this RNA message is translated into a protein. The protein, the ultimate product of genetic information, carries out the function encoded by the gene. The century of discovery up until the late 1950s illuminated the inner workings of a normal cell, but it did little to illuminate the workings of a cancer cell or the cause of cancer. Physicians had noted that some forms of cancer, such as breast, ovarian, colon, and uterine cancers, tended to run in families, and at times this was so striking that a hereditary and thus genetic cause could not be ignored. Furthermore, from, again, fruit flies, we learned that mutations occasionally occurred and that these were the results of alterations in genes, and mutations could be carried from one generation to the next. At this time, we knew radiation was known to cause cancer and induced mutations in fruit flies. So could cancer be a disease of mutations? Could genetic alterations be the unitary cause of cancer? But scientists studying viruses found that certain genes, termed oncogenes, could cause cancer. These genes encoded proteins called kinases that acted like an on switch, activating the protein's function. Often, a kinase turned on another kinase, which turned on another, and so forth, like a chain reaction. Interestingly, these viral oncogenes were just like other regulatory genes, except they were stuck in hyperdrive, activating everything around it, forcing a cell to begin dividing relentlessly. In work that has guided the direction of cancer research for the past 35 years, the current director of the National Cancer Institute, Dr. Harold Varmus, and his partner at the time, Dr. Michael Bishop, made the groundbreaking discovery that these potential oncogenes, or proto-oncogenes, exist in all of us as part of our normal cellular machinery and regulatory circuits. If mutated, these cells become what he described in his speech at the Nobel banquet as a distorted version of our normal cells. This discovery identified our genes and their susceptibility to mutation as the unifying cause of the diversity of human cancer. A second type of gene important for carcinogenesis is the opposite of an oncogene and called a tumor suppressor gene. The protein product of these genes suppresses cell proliferation. Unlike the oncogene, the tumor suppressor gene can still produce a normal inhibitory protein, even if one tumor suppressor is mutated. Therefore, it takes two insults for tumor suppressor genes to be non-functional. These findings collectively describe the core molecular aberrations of cancer cells. That is, oncogenes were activated versions of normal cellular genes. And in normal cells, they accelerate cell division when appropriate growth signals are received. But in their mutated form, they act just as a jammed accelerator would in an automobile. Tumor suppressors suppress cell division. In normal cells, they shut down cell division when appropriate signals are received. But in their mutant form, they act just as if an automobile is missing its brakes or its brakes are broken. Genes encode proteins, which often act like cellular on and off switches. Thus, conceptually, protein A turns B on, which turns C on and D off, which turns E on, and so forth. This molecular cascade is termed the signaling pathway. Such pathways are constantly active in cells, bringing signals in and out, thereby allowing a cell to function in its environment. Proto-oncogenes and tumor suppressor genes often sit at the hubs of such signaling pathways, and these are tightly regulated in normal cells. In cancer cells, activated oncogenes may chronically and permanently activate downstream hubs, resulting in uncontrolled cell division. Although dysregulated cell division is the pathological hallmark of cancer, cancer cells do not merely divide. Other activated pathways in cancer cells originating in mutated genes may block cell death or allow cells to acquire motility or increase cell survival in hostile environments or acquire its own blood supply. Abnormal genes govern nearly all aspects of cancer's behavior. These gene cascades, notably, are perversions of signaling pathways used by the body under normal circumstances. The motility genes activated by cancer cells, for instance, are the very genes that normal cells use when they require movement through the body, such as when the immune system and immune cells need to move towards sites of infection. Tumor angiogenesis exploits the same pathways that are used when blood vessels are created to heal wounds. Again, cancer is a distorted version of our normal cells. Like the earlier schematic of A to B to C of a signaling cascade, this is a more complex, yet very simplified signaling network demonstrating circuits such as motility, differentiation, viability, and proliferation with their proto-oncogenes labeled at key nodes. So how does this all relate to uterine cancer? Since the 1980s, a dualistic model of endometrial cancer development has evolved, which continues to prove useful. It was originally developed based on characteristics of the patient in cancer that could be directly observed or observed under a microscope, but these have now been augmented by sophisticated molecular characteristics as well. In type 1 endometrial cancers, estrogens are significant. Estrogens are hormones, which mean they function as signaling molecules. A signaling molecule exerts its effect by traveling through the bloodstream and interacting with cells in a variety of target tissues. The breast and uterus, which play central roles in reproduction, are two of the main targets of estrogen. In addition, estrogen molecules act on the brain, bone, liver, and heart. Estrogen receptors normally reside in the cell's nucleus along with DNA molecules. In the absence of estrogen molecules, these estrogen receptors are inactive and have no influence on DNA. But when an estrogen molecule enters a cell, passes into its nucleus, and binds to its receptor, there is a shape change. This estrogen receptor complex then binds to specific DNA sites called estrogen response elements, which are located near genes that are controlled by estrogen. After it has become attached to estrogen response elements in DNA, this estrogen receptor complex binds to coactivator proteins, and more nearby genes become active. The active genes produce molecules of messenger RNA, which guide the synthesis of proteins, and these proteins can then influence cell behavior in different ways, depending on the cell type involved. In some target tissues, the main effect of estrogen is to cause cells to grow and divide, a process called cell proliferation. In breast tissue, for example, estrogen triggers the proliferation of cells lining the milk glands, thereby preparing the breast to produce milk if the woman should become pregnant. Estrogen also promotes proliferation of the cells that form the inner lining or endometrium of the uterus, thereby preparing the uterus for possible implantation of an embryo. Paradoxically, estrogen can be both a beneficial and a harmful molecule. The main beneficial effects of estrogen include its role in programming the breast and uterus for reproduction, controlling cholesterol production in ways that limit the buildup of plaques in the coronary arteries, and preserving bone strength by helping to maintain the proper balance between bone buildup and breakdown. Unfortunately, in addition to these important beneficial effects, estrogen can also be harmful. The most serious problem arises from the ability of estrogen to promote proliferation of cells in the breast and uterus. Although this ability to stimulate cell proliferation is one of estrogen's normal roles, it can also increase a woman's chance of developing breast or uterine cancer. To review briefly, cancer is caused by DNA damage, that is, mutations, in genes that regulate cell growth and division. Some mutations are inherited, while others are caused by exposure to radiation or to mutation-inducing chemicals such as those found in cigarette smoke. Mutations also can occur spontaneously as a result of mistakes that are made when a cell duplicates its DNA molecules prior to division. When cells acquire mutations in specific genes that control proliferation, such as proto-oncogenes or tumor suppressor genes, these changes are copied with each new generation of cells. Later, more mutations in these altered cells can lead to uncontrolled proliferation and the onset of cancer. In the uterus, estrogen triggers the proliferation of endometrial lining cells during each month of the menstrual cycle, followed by death of these cells during menstruation. These repeated cycles of estrogen-induced cell division tend to increase the risk of developing cancer in two ways. Estrogen can stimulate the division of uterine cells that already have DNA mutations, and it also increases the chances of developing new, spontaneous mutations when estrogen stimulates cell proliferation. Whether the mutations are inherited or spontaneous, estrogen-driven proliferation will increase the number of these altered cells that can ultimately lead to the development of uterine cancer. One of the ways estrogen promotes cell proliferation and inhibits cell death may be through modulation of tumor suppressor function. An example is PTEN expression in normal endometrial glands, which is greatly elevated by estrogens and reduced by progestins during normal hormonal fluctuations. This reflects its role as a regulator during the estrogen-stimulated proliferative phase of the menstrual cycle. Unopposed estrogens positively select PTEN mutant cells by unmasking their inability to check the proliferative process. And this is an example of disrupting negative feedback that attenuates a proliferative signal, one of the hallmarks of cancer. Even though most cases of endometrial cancer are sporadic, about 10% of endometrial cancers have a hereditary basis. Many of these cases are associated with hereditary non-polyposis colorectal cancer, or the Lynch syndrome, a dominantly inherited syndrome with germline mutations in one of the DNA mismatch repair genes that can lead to microsatellite instability. The inactivation of mismatch repair genes disrupts DNA's proofreading and editing proteins and forms these abnormal repeat elements called microsatellites. This is an example of the enabling characteristics of genome instability through the lack of these editing systems and mutation with an increased sensitivity, leading to an increased sensitivity to mutational events and compromise of the cell's normal surveillance systems. Initial genetic changes, including PTEN inactivation and microsatellite instability, may occur in the absence of a change of histomorphology or the shape and appearance of the cell. Endometrial intraepithelial neoplasia arises through clonal expansion of these mutant cells and is characterized then by an altered cytology and architecture. The combination and order of additional mutations leading to invasive cancer differs between patients, but it may include changes in KRAS and beta-catenin or other oncogenes and tumor suppressors. Non-genetic factors such as sex hormones and sex hormone exposure may act as positive estrogens or negative progestins, selectors of mutated clones. In other words, malignant tumor develops through a series of precursor lesions accompanied by stepwise acquisition of various genetic alterations, mutation of oncogenes, tumor suppressor genes, and increased genetic instability. The type 2 endometrial cancers are usually driven by different genetic events than the type 1 cancers, but the same principles remain. P53 is a tumor suppressor gene and protein, and its mutation is one of the most common events in type 2 cancers. It is so important that it is known as the guardian of the cell, and the presence of its normal or wild-type protein prevents propagation of cells with damaged DNA. This is an example of the hallmark characteristic of cancer of evading growth suppressors. P16 is an additional tumor suppressor that is found that also regulates cell cycles. HER2/neu gene and its product is also commonly mutated in type 2 cancers, and as an activating kinase, promotes proliferation through multiple signal pathways. E-cadherin is another protein that, when mutated, acts like a tumor suppressor, as its loss leads to decreased cell cohesion and promotes motility. an example of the hallmark of activating invasion and metastases. As we learn more about the inner workings and molecular biology of cancer and these hallmarks of cancer, such as sustaining proliferative signals and evading growth suppressors, and tumor-promoting inflammation and activating mechanisms of invasion and metastases, we are also learning about key pathways that may be blocked or inhibited by specific targeted therapies. And cancer researchers are currently investigating numerous exciting opportunities to target the very things that we have learned about in the inner workings of the genetics of cancer. Much of the inspiration for the material presented today was drawn from Mukherjee's Pulitzer Prize-winning novel, The Emperor of All Maladies, a biography of cancer. This is a truly fascinating and wonderfully written story, and I've borrowed liberally from the section in this book about the origins of cancer and understanding the biology of cancer. I hope this lecture has been informative for you, and I hope you will take this back and use it in any way that you can to educate our patients and our providers better about the impact of uterine cancer and the underlying biology of uterine cancer. Again, I'd like to thank you for being invited to give this presentation and encourage you to support the Globathon, the Walk to End Women's Cancer. One step can truly make a world of difference. Thank you.